Viewers, in the series of isoenzymes, today we are going to see about hexokinase. It is an isoenzyme which has predominant role in the metabolism rather than the disease diagnosis. Let's get into the topic. Hexokinase catalyzes the conversion of glucose to glucose 6-phosphate in the presence of ATP. It exists as four forms designated from type 1 to type 4 based on the electrophoretic mobility. Type 1 has fastest mobility towards anode while type 4 has the slowest mobility. When we see the affinity for glucose for hexokinase, type 1 hexokinase has high affinity for glucose. Its Vmax is 0.1 millimolar and type 4 is around 10 millimolar. Type 1 hexokinase can be allosterically inhibited by glucose 6-phosphate and it is used to establish steady-state equilibrium in the cell. Type 4 gets saturated at a higher concentration of glucose. The glucose transporter in hepatocytes rapidly equilibrates the glucose on the cytosol and the blood. The high Km of hexokinase 4 allows its direct regulation by the level of blood glucose. When blood glucose is high after the meal, glucose is transported into the hepatocytes rapidly. Hexokinase 4 converts it rapidly inside the hepatocytes due to its high Km value. And this graph shows the saturation kinetics of hexokinase 1 and hexokinase 5. Under conditions of low blood sugar concentration in hepatocytes, glucose generated by gluconeogenesis leaves the cell before being tapped by phosphorylation. Hexokinase 4 is not inhibited by glucose 6-phosphate and hence it operates when the accumulation of glucose 6-phosphate completely inhibits hexokinase 1. Hexokinase 4 is subject to inhibition by the reversible binding of a regulatory protein specific to liver. The binding is much tighter in the presence of allosteric effector fructose 6-phosphate. This is a mechanism by which glucose is maintained by hexokinase 4. The protein anchors hexokinase 4 inside the nucleus. When increased concentration of glucose in cytosol, it equilibrates with glucose in the nucleus by transport through the nucleopores. Glucose causes dissociation of the regulatory protein and hexokinase 4 enters into the cytosol. Glucose competes with fructose 6-phosphate for binding and causes dissociation of regulatory protein from the hexokinase, relieving the inhibition. High concentration of glucose activates hexokinase 4 and during low concentration of glucose, fructose 6-phosphate triggers the inhibition of hexokinase 4 by the regulatory protein. Hence, liver does not compete with other organs for glucose. Coming to the clinical significance of hexokinase is substantial, particularly in cancer biology. Many cells show increased expression of hexokinase 2 contributing to the Warrenberg effect. The heightened glucose consumption and glycolysis observed in cancer cell. This altered metabolism supports rapid cell proliferation and survival. The mitochondrial binding of hexokinase 2 in cancer cell also helps prevent apoptosis, making it an important target for cancer therapy. In addition to cancer, hexokinase dysfunction is implicated in other diseases. Mutations in glucokinase can cause both hyperglycemia and hypoglycemia, depending on whether they decrease or increase the enzyme activity. These conditions are known as maturity onset diabetes of young, type 2 and familial hyperinsulinemic hypoglycemia respectively. Understanding hexokinase function has led to the therapeutic strategies for various metabolic disorders and cancer. Thank you for your patient listening. Let's get back with another interesting topic.